You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is December 23rd, 2024. Our topic is Abbas Chapter 11, Differentiation and Function of CD8 Positive Effector Cells. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital in Kansas City, Missouri.
This is a very short chapter, and that's the reason I typically just go over these together. Again, this is more about the same process that we just talked about, but apart from other than the CD4, now we're going to talk about the same kind of process for CD8 cells. So the chapter is Differentiation and Functions of CD8 Effector T cells, chapter 11 from Abbas. And again, today is December 23, 2024. Disclosures haven't changed in last 1 hour. And learning objectives, again, we're going to talk about the differentiation of CD8 into CTLs, effector functions of CTLs, and then role of CTLs in host defense. Couple of questions: which pair of molecules are found in CTL granules and are important for CTL killing? And then which of the following is a property of exhausted CD8 cells? I see the third question was in this chapter, not the other one. So this is another question. Naive CD8 cells, viral antigen-specific CD8 cells require activation by viral peptide bound to MHC molecules presented by APCs, but these APCs may not be infected by many types of viruses. Which of the following mechanism accounts for CD8 T cells protection against viruses that do not infect the dendritic cells? We'll come back to these. So, talking about CD8 T cells, we know that they are important for defense against the viruses as well as intracellular microbes. Some damaged cells from tumors also have a role in acute rejection of allografts. Talking about viruses, we know that they survive in a variety of cells, and even though some of the antibodies can work on the viruses when they are in their extracellular phase, when they are intracellular, there are no other killing mechanisms other than cytotoxicity. Alright. So, talking about the process of how CD8 T cells become cytotoxic T lymphocytes or CTLs. So again, talking about these naive CD8 T cells that developed in the thymus, they went through that positive selection and negative selection and became single positive CD8 naive T cells, get released from the thymus and start recirculating from one secondary lymphoid organ to another. And where if they come across the specific antigen for that CD8 T cell, then there are certain steps for activation. First is the antigen recognition itself, and then the second signal, and then some cytokines. All of that together will help in activating those CD8 T cells. Sometimes they also get help in activation from CD4 T cells. Once they do that, these cells are going to proliferate. And then during the process of differentiation, they're going to acquire all the machinery needed for killing, for their function, which is the granules. These granules are modified lysosomes that have specific functions. These cells can also secrete some cytokines, the important one being interferon gamma that helps in activating phagocytes. All right, so the important transcription factors that are present in CTLs are T-bet and eomesodermin. Looking at this figure here, we just went over this process. Here is our tissue where the infection started. Because of that, the dendritic cells or APCs got activated, got into the lymphatic vessel, came to the lymph node. Here they're gonna come interact with the naive CD8 cell, and the naive CD8 cell with MHC class 1 is gonna interact with the peptide on MHC class 1. And that interaction is gonna help in proliferation of the CD8 cells and differentiation into effector T cells. These effector T cells can then get into the circulation and go to the periphery where the infection was present, and they start helping in killing the microbes or the target cells that are infected with that virus or other intracellular microbe. Alright, so role of APCs in activation of CD8 cells. Remember, that's class 1 MHC. We were talking about class 2 MHC interacting with CD4 cells. Here, class 1 MHC uses the protein antigens from the cytoplasm of the cells on the tissue cells. There's also some cross-presentation, which means in the tissues, the class 1 MHC is on all nucleated cells. But some of these APCs also have class 1 MHC on them. There's also, if the dendritic cells or APCs are infected and they have that protein in their cytoplasm, easy enough, that's going to be taken up by class 1 MHC and going to be presented. But sometimes the dendritic cells are going to have the antigens that are going to come from outside into them. They're going to be transferred from that part to the cytoplasm, and then they go into that pathway where they're, instead of presenting on class 2 MHC, they're going to present on class 1 MHC. That's called cross-presentation. Plasmacytoid dendritic cells transfer some of these viral proteins to the cytoplasm as well. That's your cross-presentation. And then we know for cytotoxic T cells, there is some help from CD4 cells as well. So when there is a very strong innate response, severe infection that's active, CD4 help may not be needed for activation of CD8 cells. They may get activated, they may make differentiate into the CTLs by themselves. That's your severe infection. But then there are some latent infections, or maybe a transplant or tumors where the innate immune response is not as strong. In those cases, CD4 cells can help in boosting that activation. How do they help? They help by releasing some cytokines. They can also use their second signals, the CD40 ligand CD40 interaction with the CD8 cells, so that and the APCs, so that they help in activating the CD8 cells. Some of the cytokines important for CD8 cells is IL-2, IL-12, type 1 interferons, also IL-15 and 21. So IL-2 helps in differentiation of CD8 cells into CTLs, effector CTLs, or memory cells. Same thing for IL-12, type 1 interferons. IL-15 helps in survival of the memory CD8 cells, and IL-21 also induces CD8 T cell memory and prevents the exhaustion of these CD8 T cells. So what's this T cell exhaustion? When a naive T cell interacts with the peptide, say it gets activated because of a viral infection, it's going to proliferate. The cells are going to proliferate, they're activated now, and then they are going to be differentiating into these CTLs. These CTLs will help in clearing that acute infection. In that process, there might be some effector T cells, and then there are some memory T cells, and they can help in clearing the infection. But then sometimes that infection is not going to be completely cleared, and there is that chronic phase that continues. In that that time, once the CTLs have done their job or trying to do their job, they at some point are going to be exhausted, and they start expressing inhibitory receptors on their surface, because of which they are going to interact and going to be inhibited from responding to that viral infection. So some of these signals that we have already talked about are signals called PD1 on CTLs that can bind to PD1 ligand on the target cells. So that's T cell exhaustion. Let's talk about the function of these CTLs. What do? They interact with the target cell, and once they are able to recognize that target cell and make that synapse, in that immune synapse area, there will be release of these granules. That granule exocytosis causes the cell, so the synapse is formed for some time, the granule is going to be released in that synapse, and then those granules are going to be taken into the target cell. At that time, the CTL detaches from that target cell, and that target cell will continue to have those granules in them, because of which those granules will lead to the cell death of that target cell. This figure is just showing some of that interaction between CTL and target cell and that immunological synapse. Same thing in figure C. You can see that synapse between the target cell and the CTL. So let's talk about how that CTL-mediated killing occurs. So first part of that interaction is the same. There is recognition of the antigen using the antigen receptor, and within minutes, the granule proteins are delivered into that target cell. And then after that interaction, there is detachment of the CTL from the target cell, and then death of the target cell occurs in 2 to 6 hours. So we want to talk about these granules. What do they contain? So they contain specific proteins, right? So these are secretory lysosomes that are going to have these proteins, so that they are kind of in a pouch rather than being in the cell outside, because that would destroy the cell CTL itself. So these proteins include granzymes. Granzymes are different types, type A, B, C, and they have the capacity to activate caspases. Caspases, remember, are the pathway that are important for cell death. So granzymes activate caspases that lead to cell death. Apart from granzymes, there are some other proteins that are needed for the cytotoxicity of these cells, including perforin, cerglycin, and cathepsin B. There is also granulysin. So granzymes activate caspases. They are the important protein that's needed for cytotoxicity. Perforin is like complement fragment 9. It helps in making that hole in the target cell so that the other granules can be released into the target cell. Cerglycin is like the adapter protein. It just is like a base on which the granzymes and perforin can come together to perform their function. Cathepsin B is a protein that protects the CTL itself from the perforin. And then granulysin helps in altering the permeability of target cells. So all of these proteins are important for that cytotoxicity. So here there is that target cell and the CTL interacting, forming an immune synapse. The granules are going to be released in that immune synapse. At that time, the perforin is going to make a hole in the cell membrane of the target cell, and rest of these proteins are going to be released into that target cell. Then the CTL is going to be detached, and in the target cell, the granzymes are the ones that are going to activate the caspases that will lead to the cell death of that target cell, apoptosis of the target cell. Besides this mechanism, there is another mechanism that does not involve granules that's used by CTLs. That's called Fas ligand mechanism, which means so there is something called fast ligand that's expressed by CTLs. And that fast ligand can interact with fast that is a signal on the target cells. That interaction, look at that, that's a trimer. Fast and fast ligand, these are trimers. And that they have a death domain, which means that interaction leads to, again, this target cell undergoing apoptosis through caspases. So the important part to remember here is we looked at the process of how that occurs, but these granules that are formed in the CTLs can be pretty destructive, and they have to have a certain structure that in the CTL so that they can be carried from where they are formed to the surface where they're going to be released. So that structure or in the CTL is formed by microtubules inside the cell. So there is an organizing center of CTLs formed by these microtubules that move along like in the CTL so that they can take that those granules from the site of where they are formed to the surface of the cell where they are going to be released. So it's almost like a structure that's formed that's called microtubules that are going to be helping in the transportation of these granules. Once that granule is on the surface of the cell, this granule membrane is going to fuse with the plasma membrane, and so then that exocytosis can occur in the immune synapse. It's hard to understand through words, so let's look at a figure here. One of the important figures in this chapter, this is not from Abbas, but I thought this was a figure that can explain this concept pretty well. Here are the the lysosomes or modified lysosomes that have the granules. Those granules are inside that lysosome are going to move along, so they kind of have to be sorted, and then those are going to move along the cell. They have to go through these processes or steps called polarization. Then they dock onto the cell membrane. They have to be primed and then fused to the cell membrane. Once that fusion occurs, there is that hole that's created and the granules that are released, and then they enter the target cell because there is a hole that's going to be created in that cell membrane as well. And that granzyme B then leads to activation of caspases and apoptosis. So this is important because all these steps that occur have certain genes that are responsible for normal function of that step. The genes include LIST. Defect in LIST can cause so defect in any of these genes can lead to defect in cytotoxicity, but these defects can lead to familial HLH syndromes. And so some of these are really important to remember. Defect in LIST can cause Chidiac-Higashi syndrome. RAP27 is another one. MUNC13-4 is another one. STXBP2. All these genes, they can if they function well, you get normal cytotoxicity. But defect in any of these, including defect in perforin, can lead to familial HLH. Because the cytotoxicity is affected, it can cause a lot of inflammation that can be a problem. That includes Chidiac-Higashi syndrome, Griscelli syndrome, Hermansky-Pudlak syndrome, and some other familial HLH disorders, including the genes that I just mentioned, MUNC13-4, STXBP2, perforin. All of these can be defective. There are also some cytokines that I mentioned can be produced from CD8 T cells, an important one being interferon gamma and The problem with that is some of the times this can lead to pathogenesis, such as it's important in contact dermatitis, where CD8 cells secreting interferon gamma can play a role. All right. Well, I guess I talked a little bit about this. So, there are two types of infections that will lead to activation of cytotoxicity. One is intracellular organisms such as viruses that live in the cells, so they make their machinery in the cell, and they lead to those cells expressing the proteins that are made in the cytoplasm on MHC class 1 that are going to be presented to the CD8 cells, right? There are also microbes that enter from outside into the cell. But instead of being in the phagolysosome, they can actually escape from that phagolysosome and then enter the cytoplasm. So once they do that, that also needs cytotoxicity to help kill those microbes, such as Leishmania or Listeria. Defect in CD8 T cells can increase susceptibility to viral infections or intracellular microbes such as tuberculosis or malaria, latent viruses such as EBV, and then important to remember is that the cytotoxicity from CTLs can also lead to pathogenesis of certain disorders, such as say hepatitis. Hepatitis virus can live in the liver cells and they can just live there and not cause problems. But the CTLs come along and detect that virus, and the CTLs are the ones that cause the liver injury in hepatitis. There is a role of CD8 cells in tumor immunity and rejection of organ transplants, and we talked about the HLH syndromes. All right, that's it for chapter 11. A couple of questions, or sorry, 3 questions here. Which pair of molecules are found in CTL granules and are important for CTL killing of target cells?
Is this E, perforin, and granzyme B?
That is correct. Perforin and granzyme B. Perforin and Fas ligand are 2 separate mechanisms. Which of the following is a property of exhausted CD8 T cells?
Is this choice C?
Yeah,
Expression PD1 is correct. PD1 binds to PD1 ligand. And we are talking about CD8 T cells and what mechanism accounts for CD8 T cells protection against viruses that do not infect dendritic cells.
I think
I
this
think it might
is
be
C as well.
Yeah, that's what I was thinking as well.
That's correct. And that's a little kind of, important to understand that we are talking about these. I'll read the answer. So ingestion of the viral proteins into dendritic cell endosomes, and then transport of those proteins into the cytoplasm. They are processed by proteosomes into peptides that are pumped into the ER, where they bind to class 1 MHC. So instead of binding to, if they're coming into the dendritic cells, and because they are ingested, they would typically go on class 2 MHC, but that's not the point here. We are seeing how that can get into class 1 MHC pathway, and that's because of that cross presentation.
Dr. Raje, for the question number 2, I was kind of curious, for chemotherapy medications like the ones that block PD1, what effect does that have on CD8 T cells? Like, do we expect that to have a big effect on the CD8 T cells, or is it
So
kind of
there
just...
are PD1 pathway biologics that are being used right now. So they are already being used in cancers, just because they can help in taking away those CD8 T cells, activity of the CD8 T cells, and have them continue to act for a longer time.
Mm-hmm. So prevent them from being exhausted.
Correct.
Gotcha. Okay.
My understanding is, like she's saying, you activate those CD8 T cells so they can go kill cancer cells with post-exhaustion.
Sorry, I could not hear that. Can you repeat?
Never mind. I was just kind of repeating what you said, but
Well, sure. Yeah. So it's almost like, you're trying, if there is a drug that prevents the PD1 from binding to its ligand, then those CD8 T cells are going to continue to stay active for a longer time. And then they are going to fight those tumor cells for a longer time as well. And these are FDA approved inhibitors that are already being used for multiple tumors. And I believe there are more than one PD1 inhibitors that are present at this being used at this time.
Yeah, there's a lot of them. There's like almost like 8 or 9. So.
Yeah, important concept to remember. Any other questions about